Go ahead.
Thank you. Thank you very much for inviting me to share my work with you. So maybe I just start with my background. I studied mathematics and biology back in Slovakia. And then I did a PhD in computer science there. But right now or actually 5 years ago, I came to Wageningen, where I got more into the applied field of crop sciences, and now especially horticulture. So I would like to show you a little bit of type of models that I work with. And as you see on the pictures, or I don't know if you recognize it already, but I will talk a lot about tomato, but also some other plants. So first, I would like to introduce you to the concept of functional structural plant modeling. So first, some examples. The basis of these models is simulating 3D architecture and a response of plants and their growth to environmental and management conditions. So here you can, for example, see grapevine structures growing, and we can do some virtual experiments of leaf removal and see how light distribution changes and how plant reacts in growth to this. We can also test some new types of planting design. This is, for example, an apple. Does not look like it, but this is a very efficient system, very high yielding system of growing apple trees, apple fruits. Or roses in a greenhouse, we can simulate bending of shoots and growing new shoots with the flower. This is a tomato crop, and here we can see what happens if we add some lighting in between the rows. The idea is with more light, we want to increase yield. Or this is, for example, in Chinese solar greenhouse, testing the effect of planting design on the yield. And what we also look sometimes at is the difference between static simulations and dynamic. With the static, we have a crop or plant and one developmental stage, and we, for example, simulate effects of light on a given architecture, or we can have a dynamic systems where we simulate development and growth over time, like with the rows on the right. So basically, these functional structural plant models, they combine the 3D architecture, they simulate the development and the growth at a given point or over time, and in response to climate or management. I will talk mainly about the above plant architecture, so the shoot, but we can also simulate roots if it makes sense for some questions. And the predecessor of these models are process-based crop models, where they look especially on the function. So they are very good in predicting crop yield over time in different systems, crop production systems. Then in the past, structural models focus mainly on the structure without necessarily being interested in the functioning. And by combining of them, we have this functional structural plant model. So we can simulate structure, we can simulate function, and we can also simulate the responses to different, in this example, radiation levels. So let's go to tomato. I would like to give you some background. What are the parts of these models? So we start with simulating the development. So what is the architecture and how the architecture of the plant changes over time. This is often driven, this is driven by temperature. So let's say if the temperature is a little bit warmer, higher, the organs will develop at higher speed compared to colder conditions. Then once we know where the architecture is, we can calculate how much light is intercepted by the leaves of a plant and also other organs. For this, we use the information about the radiation above the crop. And from that, we know how much light is absorbed by individual leaves. And using that, we can calculate photosynthesis, the new assimilates, new sugars that the plant can use to grow. So light, but we can also take into account temperature, CO2, and other factors. And then we look how we allocate these new assimilates into individual organs. And the result is the individual organs grow and change their dimensions, their size, depending on the amount of assimilates. This is also driven by temperature. This is at one-time step, and if we repeat this cycle, the result is dynamic growth. And here is a small simulation. So starting from a seed, we can simulate then the growth of tomato plant in response to. Then I would like to go through these individual steps. So let's first look how we define the architecture and the development. So this is a 3D model of a tomato plant. In terms of topology, it's quite simple, especially if we talk about tomatoes grown in a greenhouses, because there we remove all the side shoots, and what remains is the main stem and leaves attached to it, and trusses with fruits attached to it. The leaf structure is a little bit more complex because one leaf is made of multiple leaflets, leaflet pairs, and there are some intercalary in between leaflets, between the main leaflets. So this is quite, tomato is a very interesting plant, but first we would want to simulate the shape of a whole leaf, and for that we can use some mathematical functions that describe the shape, the overall leaf shape, but we can also use it to describe the overall shape of the leaflets. Then we can look at the patterns. Okay, normally we see some main leaflet pairs, a terminal leaflet, and some leaflets in between. Now the question is how realistic we want to simulate the leaves. Is it necessary or not? So often we just take the ellipsoid or elliptic approximation of ellipse as a shape, because it was found that this is the closest in terms of light interception performance. It performs closest to the realistic shape. So sometimes it's not necessary to work with the very complex. We can use this simplification. And then we also need some information about the angle of the leaf to the stem, and of the individual segments of the leaf, of the leaflets. And when we put it all together, we have an overall leaf shape, but we also can fit individual leaflets into it. Then when we want to simulate the development, we first look what is the basic construction unit, what is repeated in a plant. So in this case, it's we call it a phytomere. It's either internode and the leaf attached to it, or internode and the flower or the truss with fruits attached to it. And then we see that there is a certain pattern. For example, in tomato, what you see is the repetition of one truss, one internode with a truss, and 3 leaves. And this basic growth unit repeats in a plant as the plant grows. And to describe it, we use L-system rules. So I will jump quickly to L-system rules. They were introduced by Aristide Lindenmayer back in '68 to study the development of algae. He was a Hungarian botanist. He also spent quite some time in the Netherlands. And basically, he observed that in a filament of algae, you see a certain types of cells that repeat. And as the organism develops, some cells create new types of cells, and you see that there is some repeating pattern in it. And formally, we can describe this pattern by using L systems as a grammar, which is defined by a set of symbols, an alphabet, axiom, an initial structure, and some rules which are applied to these symbols. So let's say we start with a cell of type A, and we have a rule that A will be replaced by A and B. That's what you see in the second row. And then we have two rules. In this case, we have a rule of how A will be replaced, how B will be replaced. And if we apply this over time, we see the development of this structure. And we can describe it in a textual form as a string of symbols. And this framework was also adapted to plants and became a very, very nice mathematical framework of plant development. So we can define some rules that will describe the development of plants over time. This textual form, for example, describes how a bush-like plant will develop, but just with the letters, it does not tell us so much. So later on, the symbols were introduced to visualize these symbols, for example, using turtle geometry. And so here we can give a geometrical representation to the symbols. We can say that F is a cylinder of a certain length, and plus is a rotation to left minus rotation to the right. And if we apply this to the rules, this will result in a nice plant-looking structure. I have one small example where I can show you step by step. So in this case, we have a symbol AB. A is a, let's say, terminal bud, B is a small lateral bud, and then F represents an internode. And we have two rules. Terminal bud will create a segment with 3 buds. Lateral bud will create a segment with a top bud. And this is how we would describe it in a textual form. And we start with the initial bud. Now we apply the first rule for A. We replace this bud by a new structure. And now we have a one terminal bud where we can apply the first rule, and 2 lateral buds where we can apply the second rule. And this is the next structure. And what is the powerful thing about this replacement is done in parallel, all at the same time, as long as the rule can be applied. So I go back to the tomato. It's relatively simple here. If the bud is vegetative, so it has no fruits, it will create an internode and a leaf as a branch, and again bud on the top. In case it creates a generative structures, flowers, it will create an internode with a flower, and this will happen over every fourth rank, and otherwise it will create leaf. So for the tomato grown in greenhouses, the rules are relatively simple. And then once we know the architecture, we can calculate how much light is absorbed. So in here, the visualization shows that the red areas would absorb the most light. For the blue areas, there is some mutual shading. They would absorb less light. And we can do this also with the crops. So we can recreate crops. We can change the planting design and see how the absorbed light changes. We can change the distances between the plants, but we can also play with the architecture. We can increase the distance between leaves and see how it affects the light and the growth. And this is done using a technique that comes from computer graphics. So it's called ray tracing. You trace rays from your light source, and then depending on the optical properties of your objects, the light rays will either reflect, transmit through the object, or get absorbed. And we can measure this with a spectrophotometer, so we can measure how much light is reflected, transmitted by the top part, top side of the leaves, or a bottom. And the last thing that is missing is the light sources. So we can use a mathematical model to describe the movement of a sun depending on the day of the year and latitude. And also we can represent the sky as a hemisphere. And if we work in horticulture, we often use artificial lighting, so we can specify the spectral properties of the lamps and also the emission pattern of the lamps. And then I will go a little bit faster through this. This would be a topic on its own, but to calculate how much sugars are created, we use photosynthesis models, and there are many of them. And basically, what they say is, in this case, what you see here is the light response. So if we increase the light, the production of assimilates increases at the beginning linearly, but at some point there is some saturation. So if you increase the light, there is not that much increase in assimilates. And we can do similar with CO2 response, a response of assimilate production to CO2. And then the last step is the calculation of the growth over time. Here we can describe this using sigmoid growth curve. So if you plot your data and look at this weight increase over time, it can be nicely described by a sigmoid type of curve. And when we then look at the derivative of the curve, we get the growth rate at each time point. And of course, there is some potential how it would look, how the individual organs would grow under optimal conditions. But in real, within the plant, there is a competition between organs, and so what you need to look at is how much sugar is available in an assimilate pool, and then what is the demand of each of the organs, and each of the organs get a fraction of what is available, depending on the competition between them. And then you can also describe the production of organs over time nicely, plant height, dry weight for organ type, and so on. So we look at the data at individual organ level, but we can also simulate well performance at the whole plant level. We can then place the plants in the greenhouse and look at planting patterns and indoor climate condition in the greenhouses and how it affects the plants. We can also do this in a smaller chambers like you would have in a vertical farming system. So this is also a tomato model, but using a dwarf cultivar. So that has a determinate growth, so it stops growing after some time. And what is very nice with these models is that you can simulate impossible. You can, for example, separate plant growth from architecture and just look at what would happen if you would modify the leaf angle of a plant. Would it respond in a better growth or not? So we can do virtual experiments, and we can try to find some idealized or optimal architecture of a plant for a given target, for a better yield, for example. And that's what we did with this dwarf tomato. So you can, for example, look at some architectural traits related to the angle, leaf angle or curvature of the leaf blade. You can look at the shape, how much leaflet shape affects the light absorption and yield, or for example, the segments, the internode length, the length between the leaves, or the segments within a leaf. And you can create ideotypes. So what you can see here is that at different planting densities, so with, for example, with 35 plants per square meter, you have each plant has a more space than, for example, with 80 plants per square meter, which is more dense. So the optimal architecture, if there is more space, would be more open. When you have less space, of course, the plant would be a little bit more compact, and this was also shown with the simulation. So things that you would expect in reality, you can also test them with simulation, but you can also go further on. And then you can see with the changes with optimal architecture for different planting densities, how it would affect light absorption and how it would affect the production of assimilates, and finally what we want to know, the yield. Also one example for cucumber. So we also grow cucumber in high wire systems, and with 3D scans we can reconstruct we can capture the architecture of cucumber leaves and plants and the architecture overall very precisely. Then we can use the point cloud data to create a plant model. So with the plant model, you can separate the points into individual leaves, petioles, or stem parts. And you can use this to reconstruct the model. You can also look at the ways of defining the leaf shape. So for example, with the cucumber, the leaves are quite curved in space, and you cannot easily use a simpler representations to describe them just with one curvature along the main axis, but they also curve along the main axis, but also to the sides. So we can use approach where we define landmarks, a certain number of points that capture the shape of cucumbers. And then we can look how the precise data from 3D scans or simplify data using these landmarks, or even such a simplification where we describe one leaf just with 2 polygons, how it affects the light architecture, light absorption. And also this helps us to decide if it's actually necessary to have such a complex shape, or if we can go on with a simplified representation. And again, we can do some virtual experiments. We can see what happens, for example, if we increase the internodes, the distance between individual leaves. If we increase it, we have some more shading at the top maybe, but what we expect is that if we increase the distance between leaves, the more light will penetrate down to the canopy. And this is what we might want to simulate. One more example with lettuce. So here, the idea is that we can scan leaves at different time points, but with scans we have a static representation. And what we would want to know is, or what we want to have is the growth over time. And that's not possible with the 3D scan. So what we did here, we tried to represent the scan using a parametric surface, using B-splines. So instead of, let's say, 40,000 points that define a point cloud of one leaf, you define a small number of control points that will describe in a parametric way this leaf shape at one time, and then you can interpolate between times, and the result is the growth like this. And you can imagine to do these models, we need a lot of data, a lot of student hands to measure individual leaves and fruits and segments, also for the photosynthesis measurement. So the last thing I would like to mention is what we did recently, and that is to use automatically collecting data like you have done in phenotyping facilities to collect automatically data and use it to update the 3D models. And you can either send your plants to the sensor, so they would travel outside of the compartment to the imaging cabinet. We would take photos of them, and then they would come back to where they grow. Or you could have sensors on top of the plants and reconstruct create the representation of the plant from the top. So we use the individual we use the imaging cabinets, the images of individual plants, and I would like to show you one example where you collect the images of the individual plants, and it's basically pixels. And what we want is to use the model. We want to use this data to update the model that calculates some outputs. So from these pixels, we first need to extract the traits, like the plant height. But the plant height is not input to the model. It's also what the model calculates. So we need to convert the plant height or the traits into the model parameters. And with this, we can update the models. And here you can, for example, see some properties of this was the 6 angles recorded of a single plant, and you can from here see what was the height over time. Now in the model, I said the height is output model of the model. We calculate it actually by looking at individual segments, individual internodes. And by summing the length of individual internodes, we get the overall plant height. And to simulate these individual internodes, we can do it with a logistic curve. So the internode elongation over time can be described by a logistic curve. Then we also know the appearance over time, which can be described by a linear curve. And this is all, these two parts are described by 4 parameters. And I just not show the details, only the results of it. So we can sense, from the images, we can get the height of individual plants over time for different cultivars. Then using statistical modeling, we can calculate what would be the average height per cultivar. And then we can use optimization, for example, Bayesian optimization, to estimate the parameters of the components for calculating the internode length. So of these 4 parameters. And this is imagined. We can also, of course, use the more detailed information from 3D point clouds. But this also needs a lot of pre-processing information. So from 3D point clouds, we can then segment or separate the leaves from the stems. And then we can calculate individual, like a branching structure of the point cloud. So individual segments, the angles between them, the angles of leaf around the plant. Of leaf is here just seen as a branch, and so we have a much more detailed information that we can use to feed the models. And this is what we use in this in the digital twin application. So all that I showed so far for the tomato was part of this project, where you have plants growing in a greenhouse. You have some sensors that record information about climate, and imaging that gives you information about the plant status. And you use this information to update the model. And when you have the model, you can calculate the output, for example, yield predictions. You can make some changes, see what happens if you change architectural parameters or planting design. And this can then be used as a decision support to make changes in the real cultivation system. So it was a lot that I showed, but to sum it up a little bit, we use this type of models, functional structural plan models, when architecture matters. For example, when we do some management strategies that affect the architecture, like the leaf pruning, or that affect the light distribution. That's what we are also often interested in. We can use it to design new ideotypes, for example, for breeding, maybe some ideotypes or some cultivars that don't exist yet, but might perform better. And one of the applications is for digital twins, for decision support. So it is not just constructed based on the climates, but also using a lot of information about the physiological processes in plants and architecture, because also growers, they often ask questions related to plant status. So are the plants producing too many leaves, or should I maybe steer climate condition that will make the plant invest less sugars into the growth of leaves, but more into the growth of fruits? And this kind of questions we can also try to answer with these models. And at the very end, I would like to show the tool that we are using. It's growing. And I just remember that we actually, my colleague back in Germany, where I worked in the past, also implemented the super shape in the tool. So I just wanted to show you some. Oh, and I also have your animation.
Nice
So
fruit.
we did it just for simulating seed shapes.
Yeah.
But yeah, that's it. Thank you for your attention.
Yeah.